Not forget, I know what we do now. So I just stand here and I use this. Okay. Okay. Sure, thank you. 3D printing is basically convert a computer model. Let's make it simple. Regardless of how complicated, as long as you can build a computer model, we call it CAD model, then this CAD model be converted into special format, it's called STL format. Then we can do that layer by layer, adding material, so that sometimes we call it additive manufacturing. We can make the structure, okay? And we are not just use that to make the machine parts. We use them to make many manufacturing design, like something we have here, and also some new product development. But 3 keywords for the 3D printing: innovation, customization, and rapid realization. So that's people interesting for 3D printing. It's just that 3 keywords and perfectly make the 3D printing technology and apply into biomedical. Why? Because we know that human beings are different and tissue are different, and it's very personalized, and we need something quick too, and if we have some problem. So that's why I get into that technology applied to bio, and we call that bio 3D printing. And the material is different. We're not just use ceramics, metal, or the plastic. We use biomaterials, and we use cells. Okay, so that's we get cell as biomaterials. We use proteins, and we even use DNA. Okay, but in my example, we don't get into that down level. We start at the cell level. But in general, this is what we defined in the bio 3D printing. Biomaterials, cells, proteins, or other biological compounds as basic building block, and to construct a personalized, the personalized is biomimic of that the physiological model, tissue model, or in vitro functional biology model. Okay, that's what we're doing. And I would like to present a bio 3D printing in 4 different levels, because people always ask, what is bio 3D printing? I say, okay, let's look at this according to how we're going to use biomaterials. Okay? If we don't care what the material you use, and we don't care the biocompatibility, that issue, then that is first level. So we don't use non-biocompatible material. Then we can use that to make medical models and medical device for surgical planning, and even for prosthesis. Okay, I have an example I want to show. But if you want to have something interface with body and input into the body, then we call it implant. Then it has to be biocompatible, but you don't have to be biodegradable. For example, hip replacement and the knees. Then, but if we want to use that as a tissue scaffold, then you have to be the biocompatible, degradable, and absorbable. Or if you want to use even something new, we call the cells. Then you get into, then make the in vitro biological models. Okay. For example, this is for the first level. Okay. And we don't have to consider biocompatibility issue. Then we use SLI to make this and determine how we can cut for the twins. This is the real case in the San Diego Children's Hospital. And how we can define the bones and tumors, and how we can define the medical, surgical planning, and how you can make the prosthesis here. And this is example of working with the doctor in Thomas Jefferson Hospital for the brain and for the electrode, and eventually make a computer model, make a 3D printed model to define how the pattern of the electrode, and how we can, this is another case, work with Children's Hospital, and how we can design and manufacture this implant and for these applications. It's not for clinical trial. Okay. Then you get into put something into the body, so we call the implants. That's got a second level. Okay, in that level, then you consider of the precisions, you consider of surface geometry, and you consider of surface function too, because this has to be deal with your cells, your host tissue. Okay. First by modeling part, the first example, the challenge is how we can make that more accurate, how complicated. Then this challenge is how surface would be, and also maybe how strong. Okay, so those are the first 2 levels. Then third level is use the tissue scaffold. This I just give a selective example, this SLA, SLS, and FDM, because I don't have time to expand it and explain every 3D printing technology details. But this is laser, this is center, laser center, this is FDM extruding. But those to show the examples of the degraded tissue scaffold. And also in Tsinghua University, we have another, the patent, the new technology. We use multiple nozzle to printing, combined PCL and TCP PLA, then makes that into this type of structure. But we make that structure into a low temperature environment. It's about negative 35 C degree. So by this phase change here, the Chinese word means phase change, then we could have a large pore with that cell to attach to grow. And also we can have the small pore size here and controllable from 5 micron to 50 micron. So in this way, we can produce, have a better bone regeneration because we can have a nutrient transfer here. This is some animal study model to show and the good applicability of this type of tissue scaffold. And we can use that technology to design those blood vessels. So this is a large blood vessel, is about 9 millimeter here, and is dog 24 weeks, and good after 6 months, we can still show the good viability of this blood vessel here. But those just the first three. But what my most interesting out of my group in last few years' work is really how we're going to printing cell. And that's what we get into. That's also because and in tissue engineering, the scaffold cell print seeding always separate. If you have a large thick scaffold, the cell is always difficult getting. And also, it's no reason why you cannot printing cells and mix cell together. So, but let's start to show what is enabling technology, and people use that to printing cell. First group of printing cell can report it. The first patent is from Clemson University, and they use HP. Okay, they just HP printer, take the cartridge out, put cells, and they call it bio-ink. Then they print it. Okay, that's Professor Thomas Boland. Later on, some companies get involved. So this is based on inkjet. Okay, they can very precisely pattern the cell, but it has difficult if you use a thermal-based inkjet, the damage the cell, and also if you want to make the volumetric, the 3D structure, so it takes time, and the cell is not that great. Second part, they call the bioplotting. The bioplotting method is more into the engineering method because nozzle people can design, whole motion platform is people designed, not like HP. HP is existing technology. So for this bioplotting method, people use, and Tsinghua University, Drexel in the U.S., and also the invention tech, that's the market sell, the single nozzle cell printing about $115,000. And Argonova, that's is RPO, the company in the U.S. Okay, they're based on use this bioplotting method, and those nozzle could be changed different size, depends on what structure you want to build. And another one is not in the commercial market. This use vibrations, acoustic droplet, and let cell pass through the passage. And also a group of people working use lasers. You have a group of cells then attached to this substrate. Okay, this laser move and controlled by the biomodel and at appropriate place and give the energy to punch the cells out and form the pattern. Okay, so based on my understanding, probably people use those one of these 4 engineer method to printing cells, as we call cell printing. Let me show you animation here, and this is what we design and patent here. We call multi-nozzle. You have to work. I don't know. It worked before I tested it. I don't know why. Let me give another try, see whether it worked. Okay. And I have to show this because I think this is important. And I know where it is here. Your mouse is not, it's not working. Oh, okay. See? Yeah. It's just animation. What I try to show you that your printed cell has to be in the sterilized environment. You can think about this in the clean room or in the hood. Then you have different nozzles. Then those nozzles could be droplet if you want to print single cells, or could be continued. And those nozzle size could be 20 micron. You want a single cell print. It could be 250, 500 micron if you want to drop a lot of cell and material. Different color here to indicate could be cells combination, different cells, cell biomaterial combination, and growth factor, and so on and so forth. It form in the 3D additive manufacture, 3D printing, starting from lines and layers and form in the 3D. Okay? So that is typical 3D printing, and just imagine all of this is the cells. And if that looks like as a cells, and this is what we printed. This is the cells. For example, we use this technology. Wait. Okay. Oh, sorry. We use this technology to pattern RGD modified the biomaterial, for example, in those green color, and unmodified, and also pattern endothelial cell and fibroblast cell. We are hoping that we use RGD, those protein growth factor, to attract those endothelial cell to form we call the blood vessel. Okay, so this is we built, and my collaborator work, and this is typical regenerative medicine example here. But what we can do really when we're printing cell, let me share with you, for example, what we do it in Tsinghua, and also my team do it at US too. And one is, that's in Tsinghua, we built a small macro liver model. It's module of a liver. Okay, it's not for function of a liver. And when we put cells, try to build this structure here, do nothing but test alcohol, because people drink alcohol and damage to liver. And we did have this good example for application for diabetes test, and we printing the fat-derived stem cells and beta cells, and we pattern and then also put insulate here. Then we compare our results and with not printing the results. What I'm showing you here is actually two different physiological models. This is what we built it. You can see endothelial cell in the outside, fat cells in the inside. This is like what a real human being. Okay, this is the fat cell is in outside, and this is not good. It's the first model. Okay, if you use this model to test the drug, particularly for undyed drug, most likely cannot get undyed. Okay, so that's maybe a reason. So this is another one example. And currently also in Tsinghua, we're working on one national key project, and we patent cells. Cell here, we are not just interested for one cancer cell, because we know all the cancer, they are heterogeneous, the heterogeneous tumor model. We try to assemble the cancer cell with the normal cell together, and not just one type of normal cell. So we try to mimic of those physiological or pathological condition here, and to build what we call stage development cancer model. Okay, my collaborator, then Professor Chen Sujun, and their team to working on this assembling model and how the cancer be developed. Okay. And this is the process. The biomaterial we use gelatin, arginate, and mix together. Then we use a cell printing method and to build a structure. Okay. And we comparing, important that is, we comparing that with the two-dimensional and with 3D model what we build, what is difference. We have good morphology to show that cancer cell is a cancer because their surround is round. But in the 2D, in the petri dishes, those HeLa cell tend to be stretched. So in the morphologic size, the cell is different. So 3D show is cancer characteristics. And also we compare the proteins, compare the drug resistance, and I don't show all the results here. But we do have one is live interview by BBC in April 11 to broadcasting this first time, and we can use 3D printing to build tumor model, called in vitro tumor model. Okay. And I also work with NASA in the US for about 5 years. to build one test drug model. You know, NASA interesting for those astronauts for Mars landing. They fly for 9 months or over 6 months. Those astronauts take medicine, we call the anti-radiation medicine. But those medicine pass liver. They know that's no problem. But they are very, very at zero gravity level. Residue of this medicine will change from EFC to HFC. How does HFC impact to the secondary organ? For example, your heart, your brain, your muscle, okay, any of that. So they call the target second organ. Okay, so that's they come to us because we have a cell printing system, and they say how we can build this macro organ system. You know, we try to build first of all, we try to build a good liver organ because those drugs have to pass through those liver. We call the sinusoidal liver organ. We patent endothelial cell and the liver cells, and build that. Then we build the heart model. So we could. This is the liver model we printed. For example, this is a heart model can print it, and we make that into very small, so 1 centimeter. This is typical. We call it tissue or organ on chip. Then at that stage, then the rest is just the NASA test study to show all these biological results, and we have 2 patents based on that too. Okay, but you know, printing cell is not that easy. So if I give you that, then that's misleading you too, and you can put all the cell together, but it's a lot of challenge, and let those cells to alive and also to function well. And I just give you feel because of time. We're not developing biology is following this way, but when we print a cell, we just use what they call the top-down approach by engineering, and we make all this structure assembling together, the cells assembling together. Okay, then we call that we build in vitro biology model. So we don't follow develop biology at a very beginning stage. So we put everything into single cell, we let it grow. Well, at the middle, suddenly give you something. So all the biology starting from here. We don't have that biology, technically. But, you know, I'm not shying to present here because he told me present something crazy. This is very crazy. But it doesn't mean that we cannot have the biology. For me, I always tell people this might be the revolutionary for biology because this is what we call the encoding the biology, new. You put everything here, not there. Okay, so that is one. You know, it's a challenge, but it's very, very exciting. Second, this is very, very multidisciplinary. It's a big field and big insight. You see, we manufacture, we use biomaterial cells, information technology, because everything you do, you have to have biomodel. You have to have a model that controls the nozzle and to tell the 3-dimensional space where the cell and how to put the cell, and also growth factor. So you design, make those macro environment and apply to life science and medicine. That's the challenge. And also, we don't have a lot of cells and materials, but that means we cannot develop. And also the structures, temperature controls, bioreactors. We talk about is not a single cell, simple culture. We talk about is a core culture, dynamic culture, even further because you build an organ. Okay. And the opportunity, I have a few slides very quick. The one is people need this. Why people need this is field is change. We all know tissue engineering. We know regenerative medicine. But that was old. That is 1989. We focused on functional tissue. But in 2007, 12 agents get together in the US. They redefine tissue engineering. That's what they define. Use physical, chemical, biological, and engineering process to control direct aggregate behavior of cell. That's called tissue engineering. It's not just functional tissue anymore. Okay? But they're adding more, they're adding the science because they think the cell biology plays a very important role. However, engineering process is key here. With that new definition, it's not just for functional tissue we have at first, it has disease model. People use that to disease and drug test model. And we have those cell tissue on chip. And today even go even far. Okay. drug test model is really people needed. We know that we have limited human can test as test sample. We don't have a lot of innovation similarity between animal model and human being. We cannot depend on 2D, that's the petri dishes, because they are all different from human. So that's why we need this, we call three-dimensional, in vitro, simulated, and this human tissue, but built in outside. Okay, so that's one. And also, that is, we are not just use this as a simple tumor, and we get this last slide. We got the human on chip. So that's, we really get into that organ printing together. But those are just for the drug test application, okay? And that's sometimes maybe you can printing a tissue structure and printing a heart. We know that may not be real printing. That's for the Xin. I want to give one. I want to have, maybe we can use this for cancer study. We build a heterogeneous cancer model, okay? We take a patient specific cancer cell out, and we use 3D printing to print the cancer. So we print it, we build this model, and we put into like a tumor on chip to test it. Okay, that's good. We can have the tools, we can have chip as product. Eventually, we try to help the doctor to determine what is good, the best way for the personalized the cancer treatment. And probably also we can help to develop what is good cancer test, the new drug. Okay. Thank you very much. Yeah.
One very quick question.